But yeah, which everything is fine. Today we'll talk about how do we color our multiple sequence alignment and what are the methods. And we will focus in this lecture about the methods that can be used with DNA sequences. As we said, there is two, we can use it for multiple sequence alignment for proteins or DNA. But today we'll only focus on this part, which is alignment coloring for DNA sequences. So we have four of them that we can use, and you will see the difference between each one of them. And we have to know when we should use any of them. The first one is called show differences. And if you just go at the arrow number one, when you open that menu, coloring menu, this will be here, the second choice here. And this is the default, as we said. So show differences coloration system is a set that set as a default, as we said. This method highlight differences observed from the consensus sequence and the alignment. And of course, you will see here, as you see, the mismatch is shown in red, as you can see here. So we are spotting the mismatches. So if there is matches, there is no problem. It will be here in the same color as gray. But if there is a mismatch, we just putting the mismatches, which is shown in red, as you can see here. So and it's like the second option here in the menu. Of course, we just enlarge this menu here and the arrow number two, just enlarge it to show that where exactly this option can be found. This and of course here's another one that you can see here's the differences in red. So this is the differences from the anchor sequence or from the main sequence that we would like to compare with. So here is the sequences that we use, and here the red color just indicate the differences from here. Okay, there is two other methods that is very close to each other. The one is called the column quality score of DNA, and the second is called frequently based differences. Both are slightly different and conceptually similar approach. So this is the first one here. You can find it in the beginning of the list on the menus of the coloration methods here at the arrow number one. And the second one is called frequency-based differences. Here you can find it at the third option here in the menu, in the drop-down menu here for the coloring. Okay, so the first one is called column quality score. The column quality score method assigns scores to nucleotides based on how well a particular base agree with the others at that column or position. So you can see how much this amino acid agree with the others. So that's why this assigned score to each one of them. This is called column quality score. So the one that most agreed is the one that can be have a higher score. The second one called frequency-based differences, and this method, the frequency-based differences method assigns scores to the bases based on their representation in the column frequency profile. Bases that occur infrequently in this are highlighted in a darker color. So you can see here color are more darker because this doesn't agree with the one that in the anchor sequence. So as you can see here, when it's far away or infrequent, it will be more darker than others. Position containing a mismatch are also highlighted in the anchor or consensus row, as you can see. So you can see here's the differences. You can spot them easily because it's just colored here, and you can see it's different from the one in the anchor sequence, and you can just put where exactly if there is a mutation or any change in your sequences. So this is a very good also method, and it is very easy to comprehend and very easy for researchers to just spot the places that if you would like to search for where is the mutation can exist and which position is mostly mutable and have a lot of much more mutation. Okay, and of course, the conserved places also you can see here, the one doesn't have the red color, this is the most conserved. So the other method called nucleic acid color. And this is easy. This coloration method provides a simple rendering for nucleic acids. So simply based on the assigning standard color. So this one, you can find it in many programs also, other than MSAV. For each base, for example, here A adenine is red and guanine, well, you will find it blue, and cytosine, you will find it in yellow, and here the green one, this is thymine. So this is very easy to spot them out, and by colors, this only 4 colors, each one for the red for the adenine, and the blue for guanine, and yellow for cytosine, and thymine green for thymine. Okay. Now we'll see another one which is called non-synonymous substitution. And this is very important, this coloring method. This is a very special coloration mode, valid only for complete CDS and alignment. If you are using or making CDS alignment, this is only very valid for that method, non-synonymous substitution. It can turn it on using of course, the URL parameter coloring equal NSS. This is called a nucleotide triplet, just a nucleotide triplet codon that are translated differently. So we just color this codon based on the translation of it. So from the consensus, you can just see the difference between this codon, the difference between different codon and the consensus sequence. From the consensus sequence or anchor sequence are marked red. So the sequences here that is translated differently from the consensus sequence or anchor sequence, like here, for example, it will be marked red, like this ones, like this ones, like this ones, like this ones here, because it will be translated differently from the consensus sequence here. So our anchor sequence are marked red. Codon are counted from the beginning of the sequence and colored in alternating gray hues. This, of course, you're talking about that ones. So it will be alternating, and the color of other ones will be alternating in gray hues, as you see here. So only the one that is different from the translated differently from the anchor sequence is colored red. So you can spot them easily from that method. I hope you can practice a little bit using this questions and quizzes here. And of course, we will continue to the next one now. Thank you. See you in the next section.